Hi, good morning and good afternoon, everyone. So we are pleased today to welcome Dorothee Dormann at the DuPont kitchen table. And so I'm just gonna introduce her a little bit. Dorothee received her PhD from Rockefeller after pursuing her postdoc at the LMU in Munich with Christian Haas. She became an independent Eminöter group leader with Michael Kiebler. Since 2021, Dorothee is now an adjunct director at the IMB in Mainz, Germany, and professor of molecular cell biology at the university there. Her research is focused on the dysregulation of phase separation, aggregation, and transport of RNA-binding proteins, such as TDP43 and FAS, in neurodegenerative diseases. More specifically, Dorothee is interested in how post-translational modifications modulate these processes and might be leveraged to suppress aberrant mechanisms. I first met Dorothee at the MPI CBG here in Dresden when she gave a talk invited by my former supervisors Simon Alberti and Tony Hyman. And I was actually blown away by her landmark study at that time she just published in Cell. She and her group had found that arginine methylation and the chaperone-like activity of the import receptor TMP01 were able to counteract phase separation and aggregation of the ALS-associated condensate protein FAS that I was also working on at the time. And yes, and now I'm super interested to hear what she will present today. Today, Dorothee will talk about aberrant phase transitions of neurodegeneration-linked RNA-binding proteins, control mechanisms, and functional consequences. Dorothee, the floor is yours.
Thanks very much, Edgar, and also to Jill for inviting me to this kitchen table talk. I'm really excited to present here and also have a nice discussion with the condensation community at the end. So feel free to ask a lot of questions, or as Jill said, also interrupt in between if you want to. So yeah, I'll start by introducing a little bit the proteins and questions that we are interested in. Try to give you a broad overview of the questions we are asking in the lab. So as was already mentioned, we are particularly interested in RNA binding proteins that aggregate in neurodegenerative disorders. And this was discovered about 15 years ago, that in ALS and frontotemporal dementia, the major deposited proteins are RNA binding proteins. And so the most prominent RBPs that we find deposited in these brain regions that degenerate are TDP43 and FAS. So yeah, as indicated here in ALS, TDP is by far the major deposited protein, and in rarer cases where you have genetic mutations in the FAS gene, then the FAS protein is affected. And a similar, you find similar scenario in frontotemporal dementia patients, where just different brain regions is affected, and therefore patients have different symptoms. So here, about 50% of the patients show this TDP aggregation, and in roughly 10% of the patients you have FAS deposited along with some other related agent RNP proteins. And just for the sake of completeness, I want to mention briefly the other proteins that you find in a subset of patients as aggregating proteins. So in ALS, this can also be SOD1, which when mutated can lead to SOD1 pathology. And in frontotemporal dementia, you also find cases that have tau aggregates. So these are the non-RNA binding protein related pathologies. And interestingly, a few years ago, it was found that also in up to 50% of Alzheimer's patients, you'll find a co-deposition of TDP along with, the more well-known neurofibrillary tangles of tau. And it was shown that also the presence of these tau aggregates in the disease, in Alzheimer's disease, correlates with a more severe neurodegeneration. So it seems to be, also contributing to the pathology of Alzheimer's disease. So therefore, it's very relevant that we understand, how these RNA binding protein deposition arises and how it actually causes disease. And this is really what we want to find out. So what has fascinated me since I started working on this about 15 years ago in the lab of Christian Haas is, as a cell biologist, I found it very puzzling that these proteins are normally predominantly found in the nucleus. And I'll show you in a minute what they do there. However, in the disease cases, in all of these diseases, you find them translocated to the cytoplasm. So the nucleus is more or less cleared. You don't find them anymore in the nucleus, and you find them deposited heavily in the cytoplasm. And so And I was immediately fascinated by finding out how this translocation and this mislocalization happens. And so this is sort of what I've been working on since. And so I'll show you on the next slide, just from a review article, what is known by now about the physiological functions of these proteins. So they basically are known to be involved in all steps of RNA metabolism. So they regulate gene expression. So from the beginning on, so from transcription, they regulate various aspects of transcription, also DNA damage repair, but then in particular also splicing regulation, also microRNA biogenesis, long non-coding RNAs, and also in the cytoplasm, where they are less prominently found, they can stabilize certain RNAs, or they can be involved in local translation, or RNA transport and local translation. So from these various functions, you can already guess that when you have this translocation from the nucleus into the cytoplasm and the cytosolic deposition, that a lot of these their normal nuclear functions don't properly work anymore, and you have a nuclear loss of function. And this is exactly what you have in these diseases. So we believe that a large proportion of the downstream mechanism, what goes wrong is the cell is really due to this nuclear loss of function and abnormal RNA processing, which occurs when these proteins get are not localized anymore in the nucleus. And also there could be sort of aberrant gain of function mechanisms from having just too much of these proteins in the cytosol, or having them deposited in a non-functional aggregated form. And so this is believed to drive the neurodegeneration in these diseases. Now, what fascinated me, as I said, and what we are now mainly working on in my group, is how this actually happening. What are the individual molecular events that lead to this mislocalization to the cytoplasm and this cytosolic deposition? We're also interested in finding out what cellular processes get disturbed when this happens, and so how does this actually cause disturbances in like cellular homeostasis and neurodegeneration. But we are mostly focusing on this aspect. So this is what most of our research projects revolve around, finding this out, both for FUS and TDP and related RNA binding proteins. So just to introduce the questions that we are asking in my lab. So I want to, step back and then sort of tell you one of the very basic fundamental, tell you about basic fundamental disease mechanisms I found back in Christian Haas's lab. And that clue about what could be going wrong in these diseases came from genetic mutations that were reported at the time. So at the time, mutations in the FUS gene that cause familial ALS were found. And you see that some of them are located in this N-terminal low complexity sequence, but a large number of them was also found in this C-terminal region, which at the time was not known what it was doing. And since it was a very arginine-rich, positively charged signal, I speculated that this could be the protein's nuclear localization signal. And went ahead and tested that hypothesis, and this turned out to be the case. So we could show back then that this is indeed the protein's nuclear localization signal. It's not a classical one, but a non-classical so-called proline tyrosine NLS, which is bound by the nuclear import receptor transpotin, or TNPO1. But when you have one of these mutations, the transpotin binding is weakened, and then the nuclear import cannot happen properly, because the transpotin 1 is one of those nuclear import receptors which belong to the importin protein family, which are known to bind to cargo proteins in the cytoplasm through an NLS sequence, and then shuttle cargoes into the nucleus. And so when this affinity is weakened, the import process is just much slower or much less efficient. And this is what is happening when you have such a point mutation. And interestingly, there were like mutations that had a different degree of severity. There were some disease-linked mutations like these ones. When we quantified this carefully, they only caused a very slight nuclear import defect. So as just seen from this, simple transfection into cells and then quantifying the nuclear versus cytoplasmic ratio. But other mutations, like, for example, this P525L mutation caused a very severe nuclear import defect and cytosolic accumulation of the Fas protein. And interestingly, there was a correlation about the reported age of onset with this cytosolic mislocalization. So back then that suggested to us that this defective nuclear import of Fas, is really disease causing. And so later on, there were even like stop mutations or frameshift mutations identified that completely truncate this NLS region, and this leads even to juvenile ALS onset. So, the more severe the nuclear import defect is, the earlier the patients get the disease. Now, what you can also see from this, and that sort of was the next question that we asked ourselves back then. So you can see that when you have such a point mutation, the Fas protein is not properly imported and accumulates in the cytoplasm, but it really sort of stays diffuse. It doesn't immediately aggregate. And so we wondered what makes it then aggregate in these patients. Why do we end up with these like large cytosolic inclusions? And we noticed back then that whenever we stressed cells that had such a cytosolic Fas accumulation due to an NLS mutation, that when we stress them, for example, with heat shock or oxidative stresses, that then the cytosolically mislocalized proteins started heavily accumulating in these clusters. And at the time, initially, we thought they are like irreversible solid aggregates, but this wasn't the case. They were actually, it was reversible when we took away the stress. And so then we, by reading more about RNP granules and so on, we figured out that this could be stress granules, which at the time were obviously already widely studied. And so there were nice marker proteins known for those stress granules, and by co-staining with various of those stress granule markers, we could show that these accumulations of Fas that we see after stress are stress granules. And so that led us to the hypothesis that when you have too much of the Fas protein in the cytoplasm due to an NLS mutation, then cells experience cellular stress and form stress granules that then Fas heavily accumulates in stress granules. And then you have a very high local concentration of the Fas protein in these granules, that those granules could really be the precursors to the pathological aggregates. And in support of that model, we collaborated with neuropathologists, with Manuela Neumann and Ian Mackenzie, and they stained brain sections of patients, FTD and ALS patients, with markers of these stress granules, like the poly-A-binding protein shown here. And they could find that a significant proportion of the Fas or TDP inclusions actually co-localize with such stress granule markers. And Ben Wollesin's group had similar findings around the same time, that there is a co-localization with TR1, for example. And so this supported the idea that stress granules could be possibly, precursors to these aggregates. Now, so this was what we knew basically, more than 10 years ago. And then at the time, phase separation or condensation was not so widely known yet in biology, but it just emerged at the time. And a few years later, basically the model emerged that stress granules would be forming through phase separation. And so then, we had this working model, and then by reading about membranous organelles and that they could be forming through phase separation, we got interested in looking into this more. And so this is obviously, to this audience, not a new thing. You're all very familiar with this concept, but this, at the time it was a new concept that became clear through work by Tony and Cliff Brangwyn and others, that stress granules are forming through phase separation. And there were observations that these physiological liquid granules can transition into a solid state. And so, this led us to speculate that these aggregates that we thought were forming through stress granules could actually be forming through such aberrant phase transitions. And so then this paper, the seminal paper from 2015 from the labs of Stephen Alberti and Tony Hyman, was published, and they used Fas as sort of a model protein to show that, this protein can form physiological phase-separated droplets at physiological concentration. So this is roughly the physiological concentration of Fas in the nucleus. So it's in the low micromolar range. And they observed in this nice in vitro reconstitution assay, as you probably all know, that over time Fas ages into these solid fibrils. And they showed that this was concentration dependent. So this is elevated or accelerated at higher concentrations. And so we figured that this could be probably, what could be going on in these stress granules, where we had shown that Fas heavily accumulates when you have too much Fas in the cytoplasm, and that this is exactly what could be going on in this process, leading to RNA binding protein aggregation. So that was really a very nice working model at the time. And so since that time, we have sort of expanded our working model, how these RNA binding protein aggregates may be forming. So we now believe that the first step is this nuclear import defect, bringing the proteins into the cytosol, having elevated cytosolic concentrations, and that can then promote their phase separation, and also obviously external stress could help to trigger that. And that then aberrant phase transitions could be going on in these granules where you have high concentrations of these proteins. And so since the time, my lab is very interested in understanding the phase separation of Fas and also TDP and related proteins better. And we are trying to figure out what are the factors that suppress this phase separation process, or what are the factors that promote it in disease. And more recently, we've also gotten very interested in thinking about what is the physiological relevance of phase separation of these proteins. Why have they, evolved to do this so efficiently? Could this have also functions in their normal, job as RNA processing regulators? Because presumably they have, adopted this behavior for a reason and for a functional reason. And so that's also something we are trying to look into now. Okay, and now I want to show you some of the major findings we had in the last years, and some of the factors we have found to suppress or promote phase separation. And how we got into how we came across one important regulator is by looking more closely at how Fas actually phase separates. So at the time, it was mainly known that this long intrinsically disordered LCD region was driving Fas phase separation, but the contribution of this C-terminal RNA binding portion wasn't looked at very much. And so we speculated, because these were also, this region apart from these folded regions is also largely disordered and has a lot of disordered RGG motifs. We speculated that this region could also be important for driving phase separation. So a PhD student in my lab at the time, Mario, he made mutations in Fas where he either deleted the C-terminal region, this NLS region, which I've previously shown you was the binding site for transportin. And then he also mutated all the arginines to lysine to test whether the arginines are important. And then I think by now this is also quite well known, that by doing such mutations, you actually severely impair Fas phase separation. So both this C-terminal RGG-rich region is important for Fas phase separation, and also the arginines, which Simon and Tony and other people at the same time had similar findings. So then after seeing this, we wondered, now if the C-terminal NLS region is so important for phase separation of Fas, could the nuclear import receptor that binds to this region be modulating phase separation? And so we tested this by adding recombinant transpotin to Fas and testing how it influences phase separation. And so we did this in very simple phase separation assays. So we always purify Fas from bacteria with an internal MBP tag. And then by liberating the MBP tag with proteases, we form these Fas droplets. Or we can also, by aging, sort of, get these more solid-like aggregate-like structures. And then when we did this in the presence of equimolar amounts of transpotin, the phase separation of Fas was completely abolished. And this was not the case for other control proteins that we added. So PRMT1 is the major arginine methyltransferase, which also binds to Fas RGG regions and methylates the protein, but that didn't have that effect. And an importin that does not interact with Fas also didn't have that effect. So we also then wanted to show this mechanism is happening in cells. And for that, we came up with a few assays. And I'm just showing for the sake of time one assay that we also now like to use a lot in my lab. And this was developed by Saskia Hutten. She adopted this assay that we usually use to study nuclear import of proteins. So here we took HeLa cells that in this case they were stressed and had stress granules, and then she semi-permeabilizes them with digitonin, and then you can wash out soluble factors and add back recombinant proteins. So you can do this to reconstitute nuclear import, but in this case we block the nuclear pores with wheat germ agglutinin, so that this import cannot happen. And when we add Fas, it actually does partition into the preformed stress granules, or it associates with these preformed stress granules. And then we asked, well, if we do this now in the presence or absence of transpartin, does this affect the stress granule partitioning of Fas? And indeed, so you see here in the top panel that Fas added alone, it nicely sticks to these preformed stress granules, which here we are co-labeling with G3BP. But if we did this in the presence of transpartin, this stress granule association does not happen. And so, yeah, we also did some assays in intact cells, where we, which I'm not showing for the time reason, because also this is all published a few years ago, and so you may anyway be familiar with it. So we could show also in intact cells that this chaperoning mechanism was happening, and that transpartin in the cytoplasm has the capacity to suppress stress granule recruitment of Fas. And now when you have what now was sort of the immediate next thought, that, well, now if you have an ALS-linked mutation in this signal that prevents or weakens transpartin binding, as I showed you in the very beginning, that now you would predict that now obviously also this chaperoning mechanism, this shielding mechanism is not working properly anymore. And this is what we could show exactly to be the case. So we showed this by purifying either the wild type protein or a protein with such an ALS-associated mutation, P525L, and then tested whether this shielding or chaperoning was happening. And you see it obviously happens very beautifully for the wild type protein, but the ALS mutant protein is sort of resistant to the chaperoning, so it cannot be properly chaperoned by transpartin because the binding cannot occur so efficiently. And this is also the case in cells. Now this is an assay where we cytosolically anchor Fas so that it cannot be imported. Basically now you can investigate its stress granule association or partitioning independent of the, its transport into the nucleus. And also here in this assay, you see that the mutant protein has a much higher tendency to partition into stress granules. And so by what these ALS-associated mutations in this NLS region are doing, they are actually doing two bad things by disrupting this transpartin binding mechanism. They not only interfere with proper nuclear import of Fas, but they also disable this shielding or chaperoning mechanism in the cytoplasm that makes then the Fas protein more prominently go into stress granules and accumulate there at even higher levels. And so that's how we think, why these mutant proteins so efficiently end up in stress granules and then can probably aggregate very efficiently there. Now this mechanism, this shielding or chaperoning mechanism is not only specific to Fas and transpartin. Around the same time, other labs showed that it was also the same for TDP-43 and its cognate import receptors. And we went on to expand this concept to other positively charged repeat proteins that arise in ALS and FTD patients. So this is a gene mutation in that you find frequently in familial ALS and FTD cases. It's a long repeat expansion in an intron of this C9orf72 gene, which was shown to give rise to a repeat RNA, which is then non-canonically translated and gives rise to this these different dipeptide repeat proteins. So if you read this repeat sequence in the 3 different reading frames, both in the sense and antisense direction, that's the protein sequences that you end up with. And so these are not made in any normal sort of, healthy individual, because they don't have this long repeat expansion. But in these patients, these proteins, these aberrant dipeptide repeat proteins are made. And so they were discovered by Dorothee and Len Petruccelli around, I guess, 10 years ago or so. And when these were sort of known to be arising in these patients, we speculated that because they're so arginine rich, they're looking a little bit like NLS sequences, that they could also be directly bound by these impotents. And Saskia tested this hypothesis and could show that this was the case. And then we and others had noticed that these very positively charged DPRs actually have the property to they very efficiently interact with negatively charged molecules like DNA or RNA, and also with low complexity sequences, for example, aromatic rich IDRs that you find in many RNA binding proteins, also in TDP. And so when you mix them together, you actually get co-condensates of this GR repeat or also PR repeat and these proteins or the nucleic acids. And then, so I'm just cutting a very long story short and sort of showing you that what now happens when you now add import receptors that directly bind to these dipeptide repeat proteins. You can do that, and by that you can actually sequester them away and sort of shield them from aberrantly interacting with these proteins and nucleic acids. And so we showed this for TDP43, so that's in this paper. And here I'm showing, so as a proof of principle, showing you that just the experiment with the RNA. So here we used fluorescently labeled poly-GR peptides that together with RNA form these RNA GR co-condensates. And this we can actually prevent efficiently by adding this important beta type import receptors, but not the, sorry, I have to move away the videos, but not the export receptors, export receptor CRIM1, for example. And so this shows that these import receptors can be, quite broadly protective and then sort of shield such positively charged phase separation prone proteins. And this concept, now really sort of has emerged from multiple other studies. As I mentioned before, Tim Shorter, back to back with us, found this was also the case for TDP43 and also other disease-linked RNA binding proteins like the HNRMP A family or the whole FET family. So their cognate import receptors can very efficiently sort of act as chaperones or even disaggregators of aggregates. And then already a long time ago, Dirk Gerlich had a similar finding for histones and ribosomal proteins, like he showed that in vitro. These also tend to aggregate with nucleic acids, with RNA, and that this can be suppressed with import receptors. And the same is true for FG-rich nuclear porins, as shown by my colleague here in Mainz, Edward Lemke, a few years ago. And so, and there is, obviously a few more other studies that I didn't have space to fit on the slide. So there's a whole, bunch of literature in the meantime that supports this concept that nuclear import receptors are broadly protective against cytoplasmic protein aggregation, especially against such positively charged DNA and RNA binding proteins that otherwise would tend to heavily aggregate in the cytoplasm. And so therefore, this concept has been proposed by us and Jim and others that possibly this could be used in new therapies if you would manage to somehow enhance the interaction of the import receptors with the pathological proteins. or elevate their levels, that could be a strategy to relatively broadly defend against such protein pathologies. Now, obviously, putting that into a therapy and into practice is obviously easier said than done, but just conceptually, I think this has emerged from this work. Now, obviously, these nuclear import receptors are not the only sort of chaperone-like or whole days type of molecules that are acting against on these RNA binding proteins. So classical chaperones or small heat shock proteins have also been shown by many different labs to be effective. And probably there could be other interacting proteins that have a similar activity. Okay, so with this, I want to leave the topic of import receptors, and for the last 10 minutes or so talk about the topic of post-translation modifications, which my lab is also very interested in. So here I'm just showing you a bunch of post-translation modifications that are commonly found on RNA binding proteins, including FAS and TDP. And yeah, these can be very complex, chain-like PTMs like ubiquitination or parylation. But what has been shown to be altered in the disease state, and that's why we have studied them, is arginine methylation of FAS and phosphorylation of TDP. And this is what I'll want to briefly talk about. So I first have a few slides on our work on FAS, because there we found, like already a number of years ago, still with Christian Haas, that FAS arginine methylation is actually altered and deficient in frontotemporal dementia. So this cartoon shows that normally FAS is arginine methylated on all these arginines in its 3 RGG domains. So this is known from a lot of methyl proteomic studies. Because we at the time found that the methylation of this signal here alters transportin binding and nuclear import of FAS, we were interested to looking into the methylation status of FAS in ALS and FTD patients. And therefore, we raised methyl-specific antibodies against this RGG3 region. And we were lucky that we got nice monoclonal antibodies that were either specific to the unmethylated RGG3 region, or monomethylated, or asymmetrically dimethylated. And then we could use those highly specific methyl-specific FAS antibodies and investigate the methylation status of FAS in ALS and FTD patients. And that showed that, so I'm not only showing you frontotemporal dementia, because in ALS we didn't find any changes, but in FTD there was a striking change in the methylation pattern. So normally healthy tissue, and this is not only true in cortex, but generally pretty much most tissues that you look at, FAS is asymmetrically dimethylated, and you get staining with this antibody, and not really any prominent staining with these other forms of these other antibodies. However, in the FTD patient cortex, where you have FAS deposited in insoluble aggregates, you find a different pattern. You don't find asymmetrically dimethylated FAS, but you find mainly unmethylated and monomethylated FAS. And so now we still don't really know exactly where this, like the switch or this deficiency is coming from. I mean, we speculate that it's probably on the level of PRMT1, and that FAS cannot be properly methylated by its major arginine methyltransferase. But, there is so far, this is more a speculation, and we don't have really proof for that. But so it's not really known how that arises. But what we sort of then looked into, what are the consequences of this when FAS loses these methyl groups? And so having shown that these arginines in these RGG regions are so important for FAS phase separation, we obviously speculate that this could affect phase separation or these phase transitions of FAS and the aggregation behavior of FAS. And so we tested this by in vitro methylating FAS. So purified recombinant FAS with PRMT1 and SAM as a methyl donor. So this worked very efficiently, and we can, like just showing you here with our specific antibodies, we can fully convert the unmethylated bacterial protein into asymmetrically dimethylated FAS. And then we could compare these different proteins, the methylated and unmethylated, in phase separation assays, or also look at the differences in dynamics in FRAP assays and so on. And that showed that, the methylated protein phase separates less efficiently and is more dynamic, and also tends to accumulate less in stress granules. So that sort of supported, that led us to the conclusion that loss of these methyl groups makes the protein phase separate more, and probably makes it more aggregation prone. Now, we are still following up what additional consequences loss of this methylation could have. And here I'm just showing you some so far unpublished data that we sort of still have to work through mechanistically. But what we also use these the unmethylated and the methylated protein for is like in vitro pull downs from just brain lysates. Where we just then, after thorough washing, sort of checked, which proteins preferentially bind to unmethylated versus methylated FAS. And there was indeed a very quite large spectrum of proteins that showed differential binding. So there were the nice, positive controls coming up. For example, transportin binds much more to the unmethylated FAS as expected, as we had previously shown. And interestingly, we found a large number of ribosomal proteins and SRSF proteins, so splicing factors, as well as translational regulators in this sort of cloud of proteins that tends to accumulate to bind more strongly to the unmethylated protein. So this is something we're still following up mechanistically. What that could mean. And then a postdoc in my lab, Aaron Sternberg, is currently looking at the consequences of FAS methylation on RNA binding. And I'm just showing you one piece of in vitro data that suggests that the unmethylated protein also has a deficiency in its interaction with RNA. So we're currently still doing sort of dynamic and affinity measurements for this. And Aaron has now also taken that into cells. And there, obviously, it's much more difficult, or it's actually not possible to really specifically manipulate or change the methylation status of FAS specifically without changing the methylation status of other proteins. But what she has done and is currently investigating is proteins where we have mutated these RGG motifs. So where we have mutated the arginine to lysine, so KGG mutant FAS, if you will. And she's now looking globally at changes to RNA binding of FAS and its RNA processing functions. And so through that, we are trying to get a handle at addressing how a FAS that shows altered methylation would show deficiencies in its RNA interactions and RNA processing functions. Okay, and so for the last few minutes, I'm leaving FAS and I want to talk a bit about TDP-43 and particularly its phosphorylation, because that has been shown already a long time ago to be altered in ALS and FTD patients. It's always been observed from the beginning on that in disease tissue where you have insoluble TDP, TDP is present as a higher migrating form. And this was shown to be hyperphosphorylated TDP. And there are also some phosphor-specific antibodies, particularly these 5 sites have been raised, and they pretty much always stain the deposited insoluble protein in patients, but not the physiological protein in sort of, under physiological conditions. And from mass spectrometry analysis of ALS brains, so there's only a couple of patients were analyzed in this study, but nevertheless, they showed that the majority of the phosphorylated sites were located in the C-terminal intrinsically disordered region of TDP. And surprisingly, it wasn't really clear what how this phosphorylation affects the aggregation status of FAS or its phase separation propensity. So there were only studies where people had globally overexpressed or inhibited various kinases that were shown to phosphorylate TDP. And this, from these studies, which are done in cell culture models or C. elegans, for example, or Drosophila, those had all come to the conclusion that phosphorylation of TDP actually promotes TDP aggregation or is sort of pathological and drives TDP pathology. And then there were a couple of studies where people had introduced phosphomimetic mutations into the C-terminal fragment of TDP, and they actually showed that this reduces aggregation in cells. And so that was really, that was really contradictory literature. And so we decided we need some in vitro studies that clearly answer this question, how phosphorylation of TDP now actually changes the phase separation propensity of TDP. And so my former PhD student Lara tackled that question by initially by in vitro phosphorylating TDP. So we found that casein kinase 1 delta, which we got from a collaborating lab in Frankfurt, can very efficiently in vitro phosphorylate our bacterial TDP protein. So you see a nice upshift of the protein. And then in simple sedimentation assays that Lara started out with, she could show that in vitro phosphorylating TDP with this kinase actually shifts it more to the soluble fraction, as you can also see from this quantification. And yeah, this was not the case, by just ATP alone or the kinase alone. So that was surprising because it, actually, we hadn't expected this finding in the beginning. So indeed, phosphorylating TDP seems to make it more soluble. And then we decided to look into this more deeply, and we made a series of phosphomimetic mutations in the sites that were found to be phosphorylated in patients. So these were either just these two aspartates, sort of serine to aspartate as phosphomimetic mutations, or these 5 ones, or 12 different ones. So all of the ones that had been found by mass spectrometry in patients. And so then various in vitro phase separation assays on these phosphomimetic proteins or control proteins, where we changed the same serines to an alanine, showed that having increased numbers of phosphomimetic mutations really suppresses phase separation of Fas. And when we looked at the morphology of the condensates that you get with the increasing numbers of phosphomimetic mutations, you see that the condensates become really much more round and large and appear more liquid-like than the relatively amorphous, not very liquid-like looking wild-type TDP condensates. And so then we wanted to confirm this by live imaging of such the different proteins, where we were looking for fusion events, and we found that pretty much the wild type protein, you almost never see fusion events happening over the time course of a minute or so. But for the phosphomimetic 12D protein, we could readily see that. And also in FRAP experiment, we found that the phosphomimetic TDP was more dynamic and more readily exchanged after half bleaches. Then Francesca Simonetti, another PhD student in my lab, set up nice aggregation assays for TDP, an STDH assay that allows us to visualize this higher molecular form, SDS insoluble aggregates. Or also we can visualize sort of insoluble TDP aggregates by confocal microscopy if we use fluorescently labeled TDP. And that also showed that having, these increasing numbers of phosphomimetic substitutions clearly suppressed this aggregate formation or higher molecular weight SDS insoluble species. And in cells, when we looked at the behavior of these proteins in cells, this really tracked with the in vitro behavior that while wild type TDP after oxidative stress, or also the 12A control protein, they readily partition into stress granules. But this was also the case for arsenide treatment, where you induce these nuclear stress bodies where TDP goes to. But then the phosphomimetic protein here, shown here, you really see that it stays dispersed and doesn't go into stress granules or into these nuclear stress bodies. Hendrik from Dieter Edbauer's lab also looked at this in primary neurons. He looked at the same panel of mutations, either like nuclear proteins or cytosolic proteins, and he did nice filter trap assays to look at the protein solubility or also the pattern you see in neurons. And that confirmed exactly what we saw in HeLa cells, phosphomimetic protein is actually much more soluble and tends to aggregate form less this SDS stable aggregates and granules basically in the neuronal cytoplasm. And so this led us to this, this is just a summary model from all of these in vitro and cellular studies that surprisingly led us to find that phosphorylation of TDP, which you do find in ALS and FTD patients, actually doesn't make the protein more aggregate more, but rather the opposite. It makes it phase separate and aggregate less. You end up with this more liquid-like dynamic behavior and really a suppression of phase separation or insolubility. And so we can clearly conclude from this, I believe, that TDP phosphorylation is definitely not a direct driver of TDP aggregation. And so we are currently trying to get more evidence for the hypothesis that this could be a protective cellular response that really is, coming up maybe when TDP starts becoming insoluble, and then you maybe have. So this is what we're currently trying to test through, again, in vitro and cellular assays. We're testing what happens when we make TDP like more solid-like versus more liquid-like. Do we actually then see changes in its levels, in its phosphorylation levels? And we're now trying to set up a cellular models of phosphorylation where we can actually study under what conditions phosphorylation of TDP actually is coming up, and where we can test this hypothesis that it's really, it could potentially be a cellular response to make TDP more more soluble and counteract the aggregation. Now, what I want to just briefly mention in the last minute is that something that we already started back then, but what is still a possibility and something we are also testing at the moment, that obviously we cannot exclude that having a more liquid-like and sort of condensation deficient TDP, what we are seeing with the phosphorylation, with the C-terminal phosphorylation, that this could actually be detrimental because maybe it could be interfering with TDP's physiological functions. And that got us now very interested in looking into the functional relevance of TDP phase separation and whether that changes its functions in RNA processing, for example. So at the time we looked a little bit into that. So this is the data that is in the paper. So we could exclude at the time that the phosphorylation or the phosphomimetic residues directly interfere with RNA binding of TDP, and also at least in a few splice targets or like auto-regulation of TDP that we looked at. This was not disturbed for the phase separation deficient 12D mutant protein, but we are now broadening this at the moment and have made now a large panel of either more phase separation prone or like phase separation deficient versions of TDP with mutations in different regions, and now are looking into how this actually disturbs TDP's ability to regulate certain physiological functions in RNA metabolism. So we want to look here at, for example, transcriptional regulation or splicing or translational regulation. So this will give us hopefully more insights into the physiological relevance of TDP phase separation. And obviously, this could also be extended to other proteins like FAS. So sorry, I forgot to advance to this slide. So this is more or less what we want to test. If we modulate TDP's ability to phase separate, how do we disturb its physiological functions that I showed you in the very beginning. Okay, so with this, I'm at the end. Thanks for sticking around. So I hope I could show you some of the current view, how we are believing these pathological RNA binding protein aggregates in neurodegenerative diseases, how they are formed. So this is our current working model. I would believe that phase separation, aberrant phase transitions, for example, in stress granules, could be one pathway how this could be happening. And I hope I could show you some of the regulatory important factors that we have found, like import receptors and PTMs. And obviously there's much more to discover. And I'm happy to go into this nice discussion with you to answer all your questions. But before I do this, I want to acknowledge my fantastic team. a lot of them moved with me to Mainz, and yeah, we settled here now very well at the Johannes Gutenberg University and the IMB. I mentioned some of the group members that did some of the studies I showed to you. And these are some of our collaborators. And yeah, I want to point out that we are still recruiting, especially postdocs, and I also have a technician position open. So yeah, if you're interested, contact me. Yeah, now I'm at the end. I'll exit my presentation, and I look forward to answering questions.
Great talk, Dorothee. Thank you very much. This was amazing. And I think we have a question from Paul Kaufmann. If you want to go ahead and ask a question.
Yes, hello.
Hi.
Great talk. I really enjoyed that. I was wondering at the very end where you showed the unaltered splicing regulation by the different mutant TDPs. Have you looked at that at a genome-wide scale or just looked at...
no, this is sort of what we are planning, yeah. And what we're now planning to do with, even more mutants, like not only this 12D versus 12A, yeah, but yeah, that is, so therefore, I think this question is still not fully answered, because in our previous study, we only looked at a few splice events. So obviously, there could be, if you look globally, there could be changes, yeah.
Great, thank you.
And also, beyond splicing, it could be in other functions, there could be deficiencies. So that's what we want to look into. But I can't answer it yet.
Great, thank you. So I actually also have a question. So it's more of a fundamental question, maybe. So do you actually believe that neurodegeneration is caused by a loss of function in the nucleus or a gain of function in the cytoplasm?
Yeah, I guess it looks, I would say, from all the data that's out there from various, for example, mouse models or neuronal cell models. I would say there is evidence that for TDP, it's really a large component of what drives neurodegeneration is the nuclear loss of function, because, for example, you see very prominent, splicing aberrations in patients as well. And so, yeah, this indicates that there is a nuclear loss of function at least happening in the disease. For FAS, there have been nice mouse model studies, for example, from Luc Dupuy's group and others that have support the idea that it's actually both, that there is also a cytosolic gain of function toxicity from having too much FAS in the cytoplasm. It has been shown that this causes, for example, translational alterations and altered interactions of the cytosolic protein. So there it looked, and that also they showed that having like basically FAS knockout is not toxic and does not drive motor neurodegeneration, but having less mutations and having too much FAS in the cytoplasm does drive neurodegeneration. So for FAS, it looks like it's an additional component of the cytosolic gain of function.
Okay.
Yeah, but in principle, both are still possibilities, I think.
Yeah. Okay, we have a question from Cameron. Do you wanna ask a question yourself, Cameron?
Hello. Yep. Thank you for the great talk, Dorothee. I was just curious. Do you find that there is any effect on the binding of the transporters when there's these different patterns of PTMs on the C terminus of these proteins? And so you see, right, the more diffuse signal in the cytoplasm, and in your summary, you also show the nucleus. But I wonder, is transport affected?
So when you have different patterns of post-translational modifications, you mean?
Yeah, exactly.
So I didn't talk about this, but for FAS, we have shown that altered methylation changes. So methylation in the RGG motifs changes the transport binding affinity. Because the transportin directly binds to RGG motifs, and then methylation status can change that. For TDP, this is still not known. And it's also not known whether the NLS region actually is abnormally post-translationally modified in the disease, although that's a possibility. So definitely, yeah, the binding, yeah, in the NLS region or around it, or yeah, can, if that's modified, this can change the affinity to the import receptors.
Thank you.
Yeah, and there could be other PTMs that we haven't discovered or studied that could also do that. That's definitely a possibility.
Okay, cool. So we have a question from Emily Spalding. You want to go ahead?
Sure, thank you. I wondered if you know whether the nuclear import receptor, TNP01, I think it was called, also regulates phase separation of wild type FUS when it's in the nucleus where it might be fairly concentrated.
Yeah, so in the nucleus, we believe that, or it's known that, the RAN GTPase displaces the binding of import receptors to their cargoes. And so, in the nucleus, this complex, by the presence of RAN GTP, gets dissociated, and therefore we believe that in the nucleus this chaperoning mechanism is not happening, or maybe then other chaperones, or the binding to the nucleic acid sort of take over that shielding or sort of holding function. So we think this is this nuclear import receptor chaperoning is more relevant to this in the cytoplasm, where you have a high affinity binding between the import receptors and the cargoes.
Okay.
Okay, then Shruti from DuPont. You want to ask your question?
Hi, thank you very much for a great talk. I just have a technical question. What was the difference between the phosphomimetic and control construct that you used for experiment?
Yeah, so as a control, we just mutated the serine to an alanine. So to a non-charged, neutral amino acid.
Okay.
So this more or less, it behaved largely similar to wild type, or it tends, like in the condensate morphology, it tends to make TDP rather a bit more amorphous or more aggregate-like looking.
Do you know why you chose alanine? Like it could be any amino acid. Yeah,
we just wanted to have something neutral, but in principle, that's why we chose alanine, and that is sort of what is typically used, as a control for this phosphomimetics, or for if you change something to a charged amino acid, then you chose something non-charged basically.
Thank you.
Yeah.
Okay, then we have a question from Katie Copley. Katie, you want to go? You're mute, Katie.
Oh, can you hear me now? Or?
Yeah. Mm-hmm.
Yes.
Okay. I was wondering whether you think, to get at the idea of if the reason we see just hyperphosphorylated TDP43 in patients is if it's effect of being in the surviving neurons, if it would be a good idea, or if you thought about doing live cell imaging of neurons and trying to track TDP43 phosphorylation and see if unphosphorylated TDP43 in a neuron, that neuron dies earlier, whereas the neurons with phosphorylated TDP43 kind of survive longer, or do you have any other ideas to try and get at?
That would be a great experiment. It's just that, so we still have to find ways to actually manipulate TDP phosphorylation in a cell model. So it's not so trivial, because as I said, if you just globally inhibit or overexpress kinases that phosphorylate TDP, that will often affect a lot of other proteins, and then it's sometimes difficult to interpret, where the effect is coming from if you see then whatever neurotoxicity or a rescue. And so that's why we so far, have only looked at phosphomimetics. And now we are looking at signals or stimuli that can actually trigger TDP phosphorylation in a cell model. So because this is actually not really known. So this is something that it looks like we have found different stressors that can do it. And so, also, I guess stresses have the disadvantage that you're also changing a lot of other things in the cell, right? And not just TDP phosphorylation. But so in principle, it would be a very great experiment to, somehow trigger, one neuron phosphorylated TDP and another neuron, have it non-phosphorylated and directly compare those. But this is technically not trivial. And so we haven't really found a way to do that.
Cool. Along those lines, I have a question as well. So do you actually know the kinase or phosphatase targeting this TDP43? Are you thinking about doing CRISPR screens or kinase glycogen kinase inhibition libraries? Or?
Yeah, so I mean, yeah, there's a few kinases reported in the literature, actually maybe a handful. This is casein kinase 1 delta, 1 epsilon, casein kinase 2, and tau tubulin kinases, and a few others. And so, yeah, we actually are considering, at least with a small panel of a small kinase inhibitor library, which we have, like looking once we have a good cell model of TDP phosphorylation, like just comparing which one seems maybe to be the most efficient in inhibiting TDP phosphorylation. Yeah, but obviously this could also all be, cell type specific, and could really be that there is a redundancy and that multiple kinases can do it. So it definitely has been shown, if you go through literature, yeah, you find, a handful or so of kinases that can, both in vitro and in cells, phosphorylate TDP. And it's not necessarily clear which one is the major, or if there is a major one in the disease setting in these patients.
Yeah, I see.
I guess, yeah, in cell models we can find out, but yeah, I cannot really answer which is the major one.
So
and I guess also same for phosphatases, like the canonical, like protein phosphatase 1 and 2 can dephosphorylate it, and I think a few others, but yeah. This is definitely, interesting to look into. But they probably broadly affect, a lot of other proteins as well. So targeting those enzymes will probably not help.
That's my related question, actually. So how would you envision drugging RNA-binding proteins, right? So are you specifically looking into such mechanisms, and like for instance, direct binders or enhancers of import activity or modulation of PTM modifiers, these kind of things? Are you
so I have to say that, with my lab, we are not doing that. I'm really like a, basically academically just interested in like basic mechanisms. So we're not directly doing that here, but obviously, I would be very thrilled if other labs sort of would maybe take up on our findings and then sort of pursue these strategies. So yeah, I guess one could, I guess for the nuclear import receptor targeting, it may be even easier to maybe do a chemical screen for finding maybe potential finding drugs that maybe enhance the binding of import receptors to these aggregating proteins or something, but it's not something that we're doing here. Yeah.
Thank you. So Jarell, Joseph, you have a question as well. You want to go ahead and ask yourself?
Hi, Dorothy. Beautiful talk. I have a question as it relates to the interplay between heat shock and the PTMs. Like, have you looked at that in the context of FOS and TDP-43, or just wanted to hear your thoughts on that.
You mean whether PTMs are changing upon heat shock? Is that what?
In terms of if you have TDP-43 or FOS that have these PTMs and you expose them to heat shock conditions, does that change the mechanisms that you see there in the absence of those stresses?
So as I mentioned, we are now looking at different stresses, like not only heat shock, but also others that can potentially, change the PTM profile of these proteins. And so, this is definitely something that I think a lot of stresses are causing changes to PTMs. I mean, although more globally, not only to those proteins specifically. But this is definitely something we're following. So I cannot really answer it yet. How exactly but that's where we would love to arrive at in a few years, maybe have a more global overview of how the whole PTM profile of these proteins changes, specifically also with different stresses. And there's definitely a possibility that this then also changes their condensation properties or general interactions in the cell a lot.
Thank you very much. Beautiful talk.
I have another question related to what we are doing at DuPont, right? So when you have a drug discovery program, you want to look at the function of the protein at some point then. Are there assays that you would actually recommend setting up when targeting those kind of complicated proteins?
Yeah, good question. Yeah, I mean, I guess the best readout for their function is, you know, to look at some of their downstream RNA processing functions, like with some splicing reporters that have been developed, or, you know, just whatever sequencing or checking for certain splicing regulatory events, which are known from large-scale transcriptomic studies or so, you know. So I guess that is a possibility. Yeah, it would be great if one, I guess I'm also very interested in establishing, or something we're working on is also establishing functional in vitro assays, where we can, you know, reconstitute some of these functions in vitro, so that we can, for example, directly with recombinant proteins, then check, you know, if we now change whatever their PTMs or their sequences, how that affects their functions. But
okay, and what do you think about the RNA transport granule function? Do you consider, like?
Yeah, I guess, I mean, yeah, I have to say, you know, we're working a lot in simple cell models, like whatever HeLa cells, and not so much in neurons, but definitely in neurons, it looks like these proteins have an important transport function, and they're present in these, you know, neuritic transport granules, and that seems to be one of their important functions. So yeah, looking for RNA transport, I mean, there's a few known targets that they are transporting, for example, along exons or dendrites. So that could, yeah, in a neuron, that could definitely also be a readout, although also obviously a bit more complicated readout.
Yeah, absolutely.
Yeah.
Well, this was
they're very multifunctional, right? So in principle, you can look at a lot of different functions, with different assays. So yeah, I think the simplest one would be a simple, you know, like there's for TDP, for example, there's some nice splicing reporters that you can just, bring into your cells. But I mean, it's obviously just tracking one particular splicing change. So, but at least it's a start for looking at, you know, complete protein deficiency or,
Yeah, thank you. Great. So Dorothee, this was great. Thank you very much. We really enjoyed it. There were a lot of questions. It was a nice discussion, absolutely, I would say.
I also enjoyed it.
And yeah, it's great to have you here. Jill, you want to say a few words?
We can't hear you, Jill.
Good.
Yeah, all right, I can hear me now. Thank you. I don't know what happened. Yes, like to thank you, echo the thank you. This was a fantastic discussion and a fantastic talk. So I think we all learned a lot and had a good time today. So thanks, Dorothee. Thanks everybody for joining on Zoom at the kitchen table at the Centrosome in our other office. And we will have more of these discussions down the line, probably in the spring. So please feel free to come back. And thanks again, Dorothee. Everybody have a good day and thanks for coming.
Thank you.
Thank you.
Bye.